Hello, my name is Caden Pate, and today we're going to talk about where did blood cells come from, how do we ID them, what is the structure of an antibody, and also what are oso, allo, and idiotypes. So getting right to it, to understand where the origin of blood cells come from, you have to understand what a stem cell is. And a stem cell is characterized by two main characteristics. It's the ability to self-renew and also the ability to differentiate into diverse cell types. Continuing, you have to understand that embryonic stem cells are pluripotent, meaning that they're going to turn into the three germ layers, the endoderm, mesoderm, and ectoderm, and those are going to give rise to our primary and secondary lymphoid organs. Our primary lymphoid organs are our bone marrow, thymus, and our secondary lymphoid organs are our spleen, lymph nodes, and sites in the gut, such as Peyer's patches. So, going back to the bone marrow, this is where our hematopoietic stem cells will arise from. And these are still stem cells, meaning that they can self-renew and also differentiate into diverse cell types. There's two different types of bone marrow. You have yellow bone marrow and red bone marrow. So the yellow bone marrow will replenish the red bone marrow. The red bone marrow will give rise to our hematopoietic stem cells. Now these stem cells will eventually become our red blood cells, white blood cells, or platelets, but they will become first our progenitor cells, and our progenitor cells are our CMP and CLP, also the CEP. But a lot of textbooks will lump these two together and call them the common myeloid erythroid progenitors. But these are no longer stem cells because they cannot self-renew. So our common myeloid erythroid progenitors will give us our granulocytes, our eosinophils, red blood cells, and our platelets. And then our common lymphoid progenitor will give us our T cells and our B cells and our natural killer cells. And it's important to make the distinction that the adaptive response will come from the CLP, and our more innate will come from some of the lymphoids, such as the natural killer, but primarily all of these monocytes and ventral granulocytes. Continuing, you have to be able to distinguish blood cells, and back in the day, people would look at them under a microscope and see how they looked under the microscope, and also how the different pH affinities and different affinities that they had for particles in the cell affected them, what color they stained them. So you have two basic dyes, well, not basic, but two dyes that you use to be able to see these things. And the first one is hematoxylin, and that's a basic dye. So it's going to bind to the acidic regions. I'm going to give a good one right here, and you can see that the hematoxylin bound to these granules, and the granules are acidic, so the basic bonds to acidic and makes a blue color. And then the eosin, which is a acidic dye, will bind to the basic particles and stain them red. So we're going to keep on going and go to flow cytometry, and this was back in the 1980s they came out with this, and it was pretty revolutionary because now we're able to look at things based on density and other factors and be able to determine what cells are what. And previously we didn't have that. Here's another picture of the way platelets look, and then you get into fluorescent microscopy, and that is a big deal because you're using live cells and able to study the function, what's going on in there. Where are you able to see these things? You would be able to put fluorescence on certain proteins and track the way they move through the cell to understand the function. And this is the best way to be able to identify certain cells' functions. But continuing, we're going to talk about the antibody structure. So you have the major classes of antibodies, which are your IgGs, which are in the blood serum, your IgEs, which are more responsible for your allergies, but they're elevated and it's not your allergy, it could be a parasite. Then you have your IgDs, which didn't know much about them, but now we kind of have an idea that they have something to do with the regulation of naive B cells. And then the IgAs, which is a dimer, and you'll see those in the secretions. And then the IgM, which is found in the blood serum, and that is a pentamer. So continuing, we get into just the basic structure, and you have a Fab region and FC region. The FC region will be your constant region, and the Fab region will be your variable region, and also where antigens will bind. So going a little bit deeper, we get into that there's variable light chains and variable heavy chains, and these places right here will make up our paratope. And our paratope is exactly where the epitope that's on the antigen will bind. And these things are held together by disulfide bridges. And let's just keep on going. So this membrane or secreted, it has something to do with it, with alternate splicing. But this alternate splicing, this post-transcriptional modification, splices out the introns and leaves the exons, and these exons will make up the gene, which will encode a protein, and depending on the arrangement of these exons will also determine what protein is being produced, so what class of antibody we're going to get, or if it's secreted or non-secreted. So these post-transcriptional modifications have a lot to do with the final result of membrane secreted, what kind of immunoglobulin. That's about all on that. But so, like I was saying, you have your antigen, and it has the epitope, and then you have the paratope, and that's going to be in the variable region. So this is a good view. These differences, IG1, IG2, IG3, and IG4, those are allotypic differences because of the FC region, but we're going to be looking at the hinged region right here. And depending on the amount of cysteine and proline residues, really determines on how flexible these different arms will be. And so it really depends on the antigen that it's trying to bind, if it's going to be profitable in the affinity of binding the antigen or not. But that's the hinged region. Now we're going to talk about the iso, allo, and idiotopes. So these are isotypic differences, but specifically a mouse's IgG will be different than a human's IgG based on these FC regions right here. And there's going to be major prominent distinctions there. And that will be the difference maker for a human's IgG or a mouse's IgG. Continuing, you have the allotypic differences like I mentioned in this slide. These are in the same class, but their FC region is different, so it's going to be an allotypic difference in that FC region. And then the idiotope. So you have an idiotope, which is on top of the variable region where you're going to end up having the binding take place. And dependent on this idiotope, it will really determine the specificity of what it's going to bind to. And once again, the epitope will go into the paratope, and these idiotopes are antigenic determinants for that. So in full circle, here's a better picture. A mouse's, this is a difference because the FC regions do vary, but in species, this IgG will differ from a human's IgG in these FC regions. The allotypic difference, we'll see these minor changes right here that are different from the major, if you notice right there. So they have the same major, but they have different minor. And the idiotopic difference will be in the variable region, in the Fab region, and it's really determined by the idiotope, and that's in the same species. And that's basically all I have to go over, and thank you.